We heard about small molecules, how you can create them, and even large molecules, biopolymers. And yeah, you can think of enumerating large libraries, and probably your drug is within this set. But there is a step in between, having the immense chemical space and the drug. And that's, in my view, or in my read, is a decision-making process, just like any other decision. You have to identify your goal, gather information to be able to avoid biases, identify alternatives, again, avoid availability bias of your ideas, weight the evidences, choose among alternatives, take the action, like select for synthesis, some of them, and then review your decision when the assay results are available and you are analyzing your SAR. So that's drug design. It's not only coming up with vivid molecules, it's about being able to collect all the necessary information and then taking the right decision. And in this, you have a loop when you evaluate the results obtained with assay measurements, and then you gather further information. And that's the field where Marvin Life comes into the picture, where you have an area, a brainstorming space, and this is coupled to an array of services that will ship you information, and these can be models, computational models, or even databases, large databases, or pre-processed data, analyzed data like MMP, which will tell you something about the SAR itself, not just the raw data itself that's sitting in your database. And on the other hand, it helps you with storing variations or even enumerating small libraries within the tool. And that's how it is in Marvin Live. You have in the middle the sketcher. You can activate plugins that's telling you some information, and you also have this idea repository. And today I would like to focus on this gather information step from all of the capabilities of Marvin Live, which is done through the plugin system. And I characterize the plugin system with three attributes. The first one is that it's service agnostic, which means that you can easily extend. It's kind of an extension point for the tool where you can utilize various services what you have available in your framework, in your software ecosystem. The plugins are real time, meaning that whenever you draw something, it will be sent to the corresponding calculator services, and then you can enjoy the results without waiting, without being disturbed in your train of thoughts. And the third one is that it's dynamic, since when you are focusing on one of the attributes, when you are balancing the attributes, drug discovery is a multi-objective optimization or a multi-parameter optimization process. However, you might not follow all of the calculated properties. You might want to focus on one of the steps, one of the things when you are doing the design, like is it novel, does it have a given liability, tox liability or whatever, or anything else. So you shift and lift your attention, and this dynamism means that you can switch on and off these plugins and have the right amount of information shipped to you through the plugins. And how we gather the information? So you have this array of services that are represented by these gears that can do data processing, pre-processed data, derived models, as I said, available in your portfolio, in your software landscape. And there are multiple ways how these can be channeled or how these utilities can be accessed in your system. One way can be that you try to reproduce it in another system, like reproducing the same database search within your system, collect the same information, or redo the models within your system. Like if you have Pipeline Pilot or KNIME, you can develop your own workflows, and then everything will be in the same software package. The other way, as Marvin Live does this, is that it offers this service agnostic integration system, so you only have to develop plugins that are calling to the array of services, and then you can utilize whatever you have within your system. And these plugins can call to REST APIs, can execute command line, can search through SQL. So there are a bunch of possibilities to be able to communicate with your existing services in your system. And what does this dynamism means? That you can select some of them and use a given subset of these plugins when you are designing. In this application study, we developed a new service, but I would like to put it into a context, how in general a calculation service looks like, what are the elements. So you have the data, you curate the data, do some magic, and this can be AI, or QSAR, or simple regression, what you like. And then you will somehow call this model, invoke, and then the plugin will show the results to the user who is the chemist and who might not be interested in the rest of this pipeline. We selected the matched molecular pair analysis since that's nowadays a standard method to dig out SAR relationship from large data sets. Matched molecular pair analysis, MMP, is shipping you results from transformations where on the same core only one particular functional group has been changed, and you collect statistics with the very same transformation across your database. So let's think of just a simple example. You change hydrogen to methyl. You have a pair of compounds different only in these two moieties, and you have a set of these pairs so that you can collect statistics that how a given property changed, whether it was beneficial or detrimental or neutral with regards to your biological activity. So essentially what you will get is a transformation, which is the primitive of an SAR relationship, and the related activity change with some nice statistics. So what you did, we used the Campbell database, applied filters on it, and then used the MMPDB algorithm. That's the selected target was HERG, which is a well-known toxicity anti-target endpoint. We only applied or model building on drug-like molecules that were standardized. This MMPA or the MMP is not available as is from the ChemAxon portfolio, but we wanted to show how flexible is this system. So we just picked the RDKit implementation of this method that's with MIT license available. That's the Hussein Ria type of MMP analysis. And the output of it is a SQLite database. And then the results were imported into JChem Web Services, and then we are running the JChem searches over it, and then the results are shown to the users through Marvin Live plugin system. So the developing plugins or connecting bits and pieces is as easy as it took 1 day to deal with that part. So from the SQLite database to be able to run the queries and show the results to the users. The harder part was this one, when we were working with all the Campbell data, when we built the MMPDB, and this was detached. That was done through Docker, in a Docker image. So that part really where ChemAxon does something. This is a generic workflow how models can be built. So what come out of this? These are the moieties. So if you have one of these in your compound that you are actually designing, we might be able to give you a possible transformation that will change the HERG related activity. And So this is the complete list, but is there a real value in this? So all of the transformations make sense? To be able to test this, we just plotted that how many of the transformations are with a given associated with a given activity change. So in the x-axis you can see the absolute median of the delta values. So the delta is in the pairs. If you go from A to B in the pairs, what's the difference in the activity? Since it can be in both directions, here we use the absolute values. So one can be positive, the other can be negative. Here you see the absolute values. So what's the change? And let's say that if it is higher than 0.3, which is these were P activity values, which is an order of 0.3 was selected as being the meaningful change in the P activity scale. So as you can see, these are the ones that might make sense. But we went a bit deeper into this and also plotted the standard deviation. So to see the statistics that if there is an effect, so you see a large effect, but what's the span? What can be beyond this activity change? So the ones with high standard deviations are cases which are not that conclusive, and the ones with lower standard deviation means that this is more conclusive. And if the absolute median delta is high enough, then you can select these cases as being the most meaningful ones, what you can really utilize during your calculation. You can find well-known pairs here, like carboxyl to ester, methyl ester. These are probably well-known examples. So how it looks in Marvin Live. This is the final plugin. That's what the user will see and what the user will face. Where whenever you do a structure and if you activate this Hög Assistant plugin, it will suggest you, it will come up as an assistant, a possible transformation by checking if from the rule set the fragment is available in your molecule, and then it will offer you a couple of transformations in an ordered way, so with the highest positive effect. So you will see first the one that will affect the most your structure, and even show you an example compound of which what will be your best choice optimizing during the optimization of that parameter, what you can easily just set back to the canvas and start working with it. So it's not just utilizing the information from the MMP, but checking if which rule can be applied to your compound you are actually working with, and then in a ranked order list, it will tell you the possible transformations. So this is what you can utilize at the end of the day from the whole Campbell Hög database. So that was about gathering information, but Marvin Live also offers views to identify the alternatives, weigh the evidence, and choose among them. I just put here a screenshot in overview mode when you would like to have an overarching view of your compound in this virtual design space, where you can assign priority, status, where you can do filtering, sorting, and rank the molecules accordingly. Or you can also visualize them in a so-called card view, in a rank ordered and filtered way as well. So that was about gathering information and about the current capabilities. And just one slide, and please reach out to András Stratz about the details where Marvin Live is going towards. It's about the hypothesis management, is about better capturing of the scientific rationale you are actually working with in a design space, and to be able to support better the review process, the decision making, even in a multi-project context. And with that, I would like to thank you for your attention.